Hi, all, and welcome to Senses Hearing Equilibrium, Chapter 16. So, it's not letting me select the thing. There it is. So, receptors for both hearing and equilibrium are located in the inner ear, and like I mentioned prior in lab, it is likely because the animals from which we evolved had inner ear structures that were important for equilibrium even back when our ancestors lived in the ocean. Those are relatively old animal structures, and then evolution, as it often does, built up on top of that a different sense using many of the same tools that were available. So there are same type of receptor cells that are used for both hearing and equilibrium, but they are in different receptor organs. The same cranial nerve carries information from both for hearing and equilibrium, although there are two branches of that cranial nerve. So hearing is a response to vibration of air molecules, while equilibrium is the body's sense of orientation, movement, and balance. The outer ear or the external ear is everything from the pinna to the tympanic membrane or your eardrum. The middle ear is where the three ossicles are, malleus, incus, and stapes, which transmit the sound to the inner ear. An inner ear includes the cochlea and vestibular apparatus and semicircular canals that are all connected to one another. So, hearing, just like seeing or sight, are describing a perception of the energy carried by sound waves. You've heard if a tree falls in the forest and no one's around to hear it, does it make a sound? And the purely scientific answer to this question is no, because sound is the perception of the movement of air molecules that happened during that fall. And in order to have perception, you have to have a cerebral cortex where perception takes place. You could say that when a tree fell down in the forest and no one was there, it created waves of molecules, but it did not make a sound. So, what is happening when we have sound? Well, here's a tuning fork that you have some experience with in lab now, and there are air molecules, right? Remember, what's in our air? There is oxygen, O2. There is some water vapor, H2O. There's a lot of nitrogen gas, which is N2. There's also some carbon dioxide, CO2, probably a bunch of pollutants and stuff. They're all bouncing around in the air. Okay. When you hit that tuning fork on your knee, it starts to vibrate. So the two sides, I guess it would be like this. Two sides of the fork are vibrating. And when they do that, they just shove all the air molecules that were near it. And they shove it kind of in waves like that, creating these refraction waves of molecules. You have visualized this by throwing a rock into a pond and seeing those ripples. That is exactly what's happening, except for it's visible in there because you're seeing those water molecules be pushed. You cannot see the air molecules be pushed like that, but it's a very similar process that is going on. Okay, and wavelength is that space from one area where you have concentrated air molecules to the beginning of the next area where you have the concentrated air molecules. Okay, so wavelength, you can visualize it on that ripples of pond in the air kind of a thing. And now let's graph it. So over time, a wavelength would be between the crest of one wave and the crest of the other wave. And there are two things that we can measure with this, and that will be intensity, which is how high these waves are, and frequency, which is how many of the waves are happening within some period of time, so maybe per second. So if it is a lower frequency, which we measure in hertz, it is a lower pitch sound. So, okay, low frequency. High frequency, do we dare? Because this is going to be awful. Ah! All right, clearly cannot sing. High pitch is high frequency, lots of wavelengths. And this, of course, is when something sounds high or low is just our cerebral interpretation of the frequency of waves. Like there's nothing about it that is high or low. It's just that we have evolved that when we have a high frequency of waves, we interpret that as a higher sound than lower frequency of wave, which we interpret as a lower sound. Okay. And then intensity, like how high these waves are, is going to be this wave amplitude, how high they are, is the loudness of the sound. So the higher the amplitude, the louder the sound, and the lower the amplitude or lower intensity is the quieter the sound. So as a review here, sound is the brain's interpretation of both frequency and amplitude, but also the duration, right? How long that horrible, unpleasant sound I made was happening. For when the waves, those ripples of air waves are hitting our eardrum, we're going to then transduce that energy pattern into action potentials that are going to interpret that as sound. So that's going to combine frequency, amplitude, and duration. As far as amplitude or how loud something is, we generally measure it in decibels. And I'm not going to go into how that measures it. We don't need to, but just for reference, low but very audible decibels is like a ticking watch. Although I don't know how many of us, you young folks, me yes, but you young folks have experienced a ticking watch sound of an analog watch, and that's pretty low. And by the time you get to normal conversational speech, so I'm probably more on the 60 decibels because I speak loudly. Live rock band, 100 decibels, that is where we begin to be in the kind of orange to red danger zone for injuring the structures of our inner ear. And you cannot see any of these really loud sounds because my face video thing is covering it, but you understand the point. A couple of things about our external ear. What's the point of it? The function is to gather sound waves and conduct them to the tympanic membrane. We need to get that sound channeled in and into toward the middle ear. We have wax or cerumen, which is produced by the modified epithelial cells of our ear canal in order to keep insects and pathogens and other damaging things from entering through to our tympanic membrane, right? We don't want it to get to the eardrum and damage the eardrum, or maybe even cross the eardrum, right? Damaging the structures. And where we hit the tympanic membrane, eardrum is where we have the boundary to the middle ear. Okay, so when the sound, I've got some sound waves here, enter the ear and strike the tympanic membrane, the tympanic membrane will oscillate at the same frequency of the waves as the sound waves were oscillating in the air itself. Okay. And then as that tympanic membrane is moving, it has the first ossicle, the malleus, attached to it, which is also attached to the incus, and then they're going to start bumping. When they start vibrating at the same what's the word I'm looking for? At the same frequency as the sound was coming in and vibrating the tympanic membrane. And so the two bones are moving like a piston, and then they tap on the stapes, that last ossicle. And this, although the sound was collected into this large area, which is the tympanic membrane, and now that all of that energy, all of it, is being passed onto a much smaller area, that means that we're really collecting and amplifying the sound. And it is now going to tap, 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 tap on the oval window that is behind this oval kind of stirrup part of the stapes. And as it's tapping, it now is transferring those waves of sound from gas-filled places into the fluid-filled spaces that are going to be inside of our inner ear. Okay, and we already talked about the structure of the inner ear quite a bit in lab. You remember that between the actual bone area and the tubes, there is a little bit of space, and that is the space that's called, that's filled with perilymph. And then you actually have some tubes inside of that perilymph, which is inside of the bony labyrinth. And those tubes are themselves filled in with endolymph. So again, we're taking that energy from the waves of sound and transferring it from gas-filled chambers to fluid-filled chambers. So first we'll look at the cochlea, so that's the snail-looking part of the inner ear, which is where the sound transduction is occurring. So remember, transduction is that process of taking a certain form of energy. In this case, it's going to be sound energy, the vibrations of these molecules as they're bumping in this rhythmic way, and we're going to change that form of energy into action potentials, and which is going to be this electric current that is going to be sent to be interpreted in our brain. Okay, so I showed this to you already, showing that if you were to uncoil that cochlea, just to see kind of what the structure looks like, because the coiling makes it pretty confusing to figure out, you will see that all of the perilymph is totally connected. Okay, and the this little point area, which is the kind of end of the snail shell shape, is where the perilymph can now get bumped and be like imagine that you're pushing fluid, right? And if you had nowhere for it for that energy to go, then it could possibly like pop, because right, if you're pushing the fluid and it goes and it hits against this part of the structure, then it could break stuff, because fluid is not compressible. Water or general aqueous solutions are not compressible. Liquids are not compressible very much. So when you move it, you're displacing it. So when the stapes, right, that last small ossicle, is bumping on the oval window right here, it pushes that perilymph. When it pushes that perilymph, it flows in those waves down and around, and then pushes on the round window. And so the energy is eventually, it's interpreted as it's flowing through, and then the end of it is absorbed by the round window, so that you do not have, like, this is the place where you can displace some of that liquid, where it has a flexible area to kind of be pushed into, and then it'll eventually come back. Okay. The cochlear duct, which is also called scala media or scala media, is separated from the other two ducts, and it's filled with a different fluid called the endolymph, endo inside. And endolymph is strange because even though it is outside of cells, right, this is extracellular space in this area. There are cells up here, and then there are cells right here making the boundaries of these tubes. The space in between is extracellular space, but the composition of the endolymph really resembles intracellular fluid. So it's really high in potassium and low in sodium. It's not exactly like intracellular fluid, but it is similar. So that is a strange thing for inner ear in comparison to every other place we have learned. Organ of Corti, which is also called the spiral organ, and I should have put that here, sits on the basilar membrane of the scala media or the cochlear duct. And this organ right here, sitting on the basilar membrane, it is also partially covered by this kind of gooey membrane called the tectorial membrane. And the actual hearing cells are called the hair cells, and they have these stereocilia. They're not true cilia, but these projections that we call hairs, and they are embedded into that tectorial membrane. There are also some supporting cells. There are different types of hair cells, and I'm not really going to go into that very much. And of course, how do we know which ones are the hearing cells other than the hairs that are sticking out? And it's small to see here, but there are direct connections with dendritic endings. So they're represented here in yellow, neural tissue, neurons. So as we're going to look at the mechanism of the physiology of hearing, let's look at a model that is not as complicated as an ear, but gives you a mechanical understanding of what's going on. So we're getting these sound waves. They're hitting on the tympanic membrane, which is moving those ossicles in the air. And finally, the stapes here is pounding on the oval window. When it's pounding on the oval window, it is pushing that perilymph, that fluid, boom, 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 like waves, right? Well, unlike air, fluids, including perilymph, is not compressible. So it's like in a pool, you can't, or in your bathtub, when you're pushing on the water, it's not just going to like just take less space. It's going to bounce out of the pool or out of the tub because it still needs to take as much space as it did before. And when you've shoved it, that means the end of it also needs to be shoved somewhere. Okay, so that means that as the stapes is pounding, it's pushing on that perilymph is being displaced, and as it's being displaced, it is pressing on the perilymph forward, but it's also pressing on the basilar membrane that is right right there, right? Because it's not really pushing in a linear direction, it's pushing just out and in all directions from it. And as it's pushing on this basilar membrane, it bends, and that means that the hair cells are going to be bumping with it. Well, that perilymph is going to get shoved, go all the way around. Perilymph, remember, it's all connected. It's going to keep going around until bloop, it pushes onto the round window. And the round window just has this flexible bit of epithelial cells, so they just bloop, they just push in. And then after they push in, then they'll push out. And when they push out, the whole thing goes in reverse because all the fluid is now bouncing in reverse. So that means as the stapes goes in, out, in, out, in, the tympanic membrane of the inner ear is going out, right? This oval window area is going out, in, out. So the opposite of what the stapes. And as that is happening, the basilar membrane and therefore the hair cells that are attached to it are going down, up, down. So I hope you can picture how this is happening, and mechanically it's not super complicated. What's hard to imagine is that can happen up to 20,000 times per second. So let's look at what's going on in an actual inner ear. Here we have all of the structures that we've already talked about. The only other thing I want to bring attention to here is, let me get a little pointer thingy. Hey, pointer. Is the tectorial membrane. The edge of the tectorial membrane is anchored to like the inner kind of around the axis onto which the cochlea is coiled, that inner core of the cochlea, that is where the tectorial membrane is attached, and it's quite stiff. So it's basically, as everything's bouncing around, it remains unmovable because it's in the center and everything's flowing around it, so it's not bumping it into any direction. So as vibrations are sent through the perilymph, they move all the perilymph in every direction, and this causes the basilar membrane to bounce up and down, and the hair cells are dancing with it because they are attached. But because the tectorial membrane stays intact, the hair cells are moving against it. So they're being pushed one direction and then they go the other direction. So that tectorial membrane is important to have there because it's the only thing that is stationary, and so the hair cells can be pushed against it. Okay, we'll see why that's really important in a second. And the hair cells are mechanoreceptors. What does that mean? That means that the stretching of the plasma membrane of these cells is the thing that is going to open in this case potassium, but it's going to open some sort of an ion channel that's going to cause change in voltage because of the flow of ions and a action, or in this case, a depolarization that will lead to an action potential. I will show you in just a moment. Okay. So as you have seen, these hair cells are not themselves, strictly speaking, neurons. Therefore, when this movement is happening and the hair cells are like bending against the tectorial membrane and therefore stretching their own plasma membranes and causing the distortion of that plasma membrane and opening of the ion channels, when the actual hair cell depolarizes, it, through multiple steps that I'm not going to go into, causes a transmitter, like a neurotransmitter, to be secreted by exocytosis from this end part of the hair cell, and those transmitters are going to bind to the plasma membrane of these neurons, and then the neurons are going to fire an action potential, sending that message to our brain. So here, let's review one more time. Sound waves strike the tympanic membrane, and the sound waves become vibrations. So that's still outer ear. The sound wave energy is transferred to the 3 ossicles of the inner ear, which vibrate. Now, we're still pushing that energy through air. Now, in the middle ear, the stapes in the middle ear, that last ossicle, is attached to the membrane of the oval window, and it taps on it. And the vibrations of the tapping on the oval window create fluid waves. So now we're transmitting those waves into a fluid here in the perilymph within the cochlea. The fluid waves push on the flexible membrane of the cochlear duct. And the hair cells bend and ion channels open, creating an electrical signal that alters neurotransmitter release. Neurotransmitter or transmitter, depending on how strict we want to be with our terminology. This transmitter or neurotransmitter is released onto sensory neurons and create action potentials that travel through the cochlear nerve to the brain. Energy from waves transfers across the cochlear duct to the tympanic duct, right? This tympanic duct here. And it dissipates back the bloop into, okay, Spartak, come on, to the middle ear on the round window. So that fluid displacement has to go somewhere, and that is onto the round window. Okay, so here if we're looking a little bit more deeper into it, we have these hair cells. They vary in size, they're connected to one another, and they have a mechanically gated ion channel. And notice how cool it is that it's closed, and then as the membrane, as these top links, these are proteins, push. So we have fluid that's moving right here, moving, and it pushes on this tallest one, which bends, and because it's connected to all of its neighbors, they're going to bend, and that opens the mechanically gated channel. And remember that endolymph has this strange concoction of ions, including a really high concentration of potassium, which means that there is actually a gradient for potassium. So when the graded channel opens, it is potassium ions that flow into the hair cells. This is different than anything we've talked about before. Potassium ions are the ones that are flowing in because there is both an electrical and a concentration gradient to drive this ion inside. And as it rushes in, it depolarizes that hair cell. That depolarization ends up causing other ion movement, and eventually vesicles that have the transmitter packaged in them gets secreted due to the change in ion concentration and voltage. And when the exocytose, those transmitters bind to the actual neurons, opening those ion channels, which causes depolarization of the neuron. And if it's big enough, if the graded potentials add up to be big enough, it will send an action potential. Yay. So how do we really discriminate the location, the pitch, the loudness of the sound? There's a lot that goes into it, and I'm trying to keep it incredibly brief, so I'm going to just give you a tiny bit about that. The sound processing first happens in the cochlea, where we can determine the pitch of the sound. Remember, pitch is that horribly high out of tune note that I had versus the low note that was slightly less out of tune. The basilar membrane, remember basilar membrane onto which all those hair cells are stuck? the basilar membrane, which is part of the cochlear duct or scala media, it is not consistent throughout the entire cochlea. The closer it is to the middle ear, to basically the beginning of the path, the stiffer it is. Stiff and narrow, where it's attached to the oval window and the round window by the middle ear. But as you go further away from that, it becomes more flexible and thinner at the distal end. So when you have high frequency waves, there's enough energy there to cause the vibration near its base. And the way our brain interprets that is that if action potentials are being sent by cells that are close to the base of the basilar membrane, that is interpreted in our brain as a high-pitched sound. Low frequency waves, they travel along the basilar membrane and they are not strong enough in energy to cause vibration at the stiff section of the basilar membrane. They only cause vibration at the distal, more flexible end of the basilar membrane. And if our brain is receiving information from the neurons associated with the hair cells at that distal end of basilar membrane, then we interpret that as low pitch sound. So think of it as like if you were to unfold the whole cochlea like we've seen before, that is like a little keyboard. And just like if you hit the keys on one side of the keyboard, it's going to have lower pitch, and on the other side of the keyboard, it's going to have higher pitch. And I think I'm mirrored in this camera, but I do know that it's on the left, that is the lower keys on the piano, and on the right, that is the high keys. There you go. Okay. Here is, I should have moved the picture over, but you can see that we're trying to tell you that the basilar membrane is stiff over here, and so it responds better to the higher energy, high frequency waves, which are high pitch, and at the apex, low energy, low frequency. And in the middle, it is medium frequency wavelengths. So again, just like we were talking about our general senses and talking about the mapping of your primary somatosensory cortex in that postcentral gyrus. Remember the picture I had of the gyrus here, and if this cell in that postcentral gyrus is receiving action potential, it interprets that you've been touched in your finger as opposed to on your nose, right? Similar, again, it's the location in which it's being stimulated is interpreted differently. But in this case, instead of at the end of the whole pathway that we have that mapping, we have the mapping here at the beginning of the pathway, or at least this is one part of how we interpret the sounds. Loudness of sound is coded the same as in all of our somatic receptors. The louder the noise, the higher the frequency of action potential. So if you have more action potentials per second, we perceive that as louder, and lower frequency of action potential is perceived as quieter. And how do we know where the sound is coming from? You probably already have guessed this. That's helpful that we have two ears, and we are able to process the timing difference of hearing something from one ear versus the other ear. So you have a separate pathway into your cerebrum of hearing, perceiving the sound from one ear and the second ear, and then there's a whole calculation, more axons that communicate and calculate the time difference. Which one came first? Which one came second? How big was the difference in that timing of the arrival of action potential to your cerebrum? And we perceive that as localization of sound. So unless sound is coming from exactly in front of the person, it will not reach your ears, both ears at the same time. And the brain records the time difference for sound arriving at the ears and uses very complex calculation to create a three-dimensional representation of the space around you and the sound source within it. You don't need to see the key, do you? So here we have a sound wave is coming. It's going to hit one ear or the other, and the cochlear branch of the vestibulocochlear nerve, cranial nerve 8, is going to go, and half of it is going to decussate while the other half is going to continue on. We're also going to have some collaterals, some branching of this move to the cerebellum, because interpreting different sounds is important in being able to stay balanced. And then this information will travel to the thalamus and to our auditory cortex. Okay, the auditory cortex is here in the superior part of your temporal lobe. Okay. Some people are extremely good at being able to localize sound. And this is especially true for people who have lost their sight. There is an amazing Radiolab episode about this from years and years ago, back in the old school classic Radiolabs that I loved so much. And Radiolab used to be a radio show that then became a podcast, and it still is, but it's now pretty different than it was before. And it's talking about using echolocation. Literally, people who are blind some people can do these little clicks. They're not super loud, but they're clicks that are audible. And from how the sound bounces off of objects around them, they can basically build a three-dimensional perception of the space around them. And some of these folks who are completely blind, can ride bicycles and walk without canes or without guide dogs extremely well. It's pretty amazing. Not everybody's brain is set up in a way that they can do that if they go blind, but some people can. And it's just an extremely full use of the system that we have evolved for localization. All right, so hair cells release transmitter or neurotransmitter onto bipolar sensory neurons. The first order neurons travel through the cochlear branch of cranial nerve 8, to the cochlear nuclei of the medulla. Second order fibers from cochlear nuclei of the medulla send signals either to the superior olivary nuclei of the pons or the inferior colliculi of midbrain. Remember colliculi that you learned in the first lab, those 4 bumps on the back? The top 2 superior colliculi were for vision processing, and the bottom 2 were for auditory processing. This is really important for the startle reflex. So if you hear a sound that startles you, we have a reflex to turn toward that sound. Right? And it's also important in sound localization. The third order fibers from the inferior colliculi travel to the thalamus. And then there is here a fourth order fiber that project to the primary auditory cortex on the superior temporal lobe. So there are 4 neurons that are involved instead of 3. Okay, I'm going to talk just a little bit about the physiology of equilibrium. We talked about the vestibular apparatus. Right? Vestibular apparatus contains the cristae and maculae, which are organs for equilibrium. Okay. And one of them is in the saccule and one of them is in the utricle or utricle. There are also 3 semicircular ducts in the three semicircular canals. And there are three kind of enlargements of the semicircular canals called ampullae. That's the plural. Think of enlargements for each of the semicircular ducts is like a ampulla is a little bottle. So in the saccule and utricle of the vestibular apparatus, we have the organs that provide you information about the position of your head in relation to gravity, right? So if I do this and then back like this, my brain is interpreting that through those two maculae. And also information for linear acceleration or deceleration, so how fast you're going forward and back. Okay. In those ampullae of the semicircular ducts, we have information about rotation of the head at different angles, and all of that information converges together and gets calculated by your brain to be able to tell which way you're spinning. Okay. Very, very similar to what we have already learned, there are hair cells in the saccule and utricle of vestibule, and the organs that hold that is called a macula. Okay, we have two of them. We already talked about the fact that the hair cells of the macula are covered in otolithic membrane that there's a gelatinous substance, and then otoliths, they're actual crystals that are on top of that. They're attached to that gelatinous substance. And when we have linear acceleration or movement of ear, these otoliths shift due to gravity, direction of the gravitational pull being changed. Well, maybe not. So in this case, it would be directional pull, but in the case of linear acceleration, it's the kind of momentum buildup in how those otoliths move. And that pulls on the cilia of those hair cells. And again, mechanically gated ion channels open, ions flow in, and the same thing happens that we already learned in the, what's the other stuff? In the organ of Corti, or of the cochlea. Okay, so it's just showing you here the gravitational force pulling on those otoliths, which are attached to the gelatinous membrane, which are attached or have in them embedded the hair cells, therefore stretching the plasma membrane of the hair cells. Okay, similar thing is happening in each ampulla of the three semicircular ducts. The organ that they have in there is called a crista ampullaris, or just a crista, and they have hair cells that are going to detect rotation of the head. We have the three different ducts arranged at different angles in order to detect three different planes. We talked about this already. The gelatinous cover of the hair cells in the ampullae is called cupula, and this cupula is gelatinous. There are no otoliths, so no rocks in the semicircular ducts, no rocks involved here. And again, the same thing when we have a movement of the fluid in there. Let's look at a picture here. When we have the movement of the endolymph, the fluid moves faster than, right, it gets this momentum, and then it pushes on the cupula, that gelatinous stuff that's covering the hair cells. And then when you stop, there's a delay, but then eventually the fluid goes back and the cupula is pulled back, and therefore the hair cells will move into their original position and the mechanically created channels will stop. being opened. All right. So eventually that those hair cells will become cats around me, all around me. Those hair cells will secrete a transmitter or a neurotransmitter onto the neurons that are near them. Those neurons will depolarize and potentially send an action potential to the brain via first order fibers. So they will then travel through the vestibular branch of the vestibular cochlear nerve, cranial nerve number 8, to vestibular nuclei of pons and medulla. The eyes and other proprioceptors, right, in our joints provide sensory information that will be combined with what we're getting from our inner ear, thus giving you your overall understanding of your position in space and of your movement. And if that what you're seeing through your eyes does not match what you're getting from your inner ear or from some of your other proprioceptors, then your brain can freak out and say, okay, something doesn't add up, it doesn't match. I'm just going to make you feel sick. So you stop doing whatever this crazy thing that you are doing, like riding in a car. So cerebellum is going to receive some information from your vestibular apparatus. You're going to then get some information is going to go to the nuclei from where these cranial nerves begin for eye movement. So you'll get information over there so you can react with your motor neurons appropriately. Reticular formation, if you remember, that's a cluster of nuclei in your brain stem that are going to adjust your respiratory rate and blood circulation and everything when your position changes, so you're appropriate. Right? Some reflexes may be activated at this point, allowing you to maintain balance. Okay, so you may need to shift some weight, use that knee-jerk reflex to actually balance you. And the thalamus will filter some of these signals and let them continue on to the primary sensory cortex for perception of your position and movement. Oops. So this kind of outlines all of those pathways, and of course this is very simplified version. There's a lot more going on, but it still looks quite complex. And in order to know what it is that you need to know for your exam, please check out the study guide, and I will see you later. Oh, never mind.